Have you ever opened the Seurat tutorial, looked at it for a second, and quietly closed that tab? Well, this video is for you. I'm Pablo Farias, and in this video we're gonna go over the official starting guide for Seurat. Now, full disclosure, I have been building software most of my life. I build web applications, games, AI agents, lots of cool stuff, but I've only started learning biology in the last couple of years. It's become an obsession of mine. And it's only been recently that I started bridging the world of biology with software by teaching myself bioinformatics. So please let me know in the comments if I make any mistakes, if you have any feedback or suggestions. So with that said, let's get started. The first thing we'll need is to download and install R and RStudio. I wasn't sure if I actually needed RTools, so I installed it anyway. You might have to install it, so have a look at that. If you run into any errors, you might need to install RStudio as well. The tutorial that everybody points on the official Seurat website, at least at the time of this recording, is actually not the latest one. So this is not using Seurat 5. This is the old version of the tutorial. You can find the new version of the tutorial on GitHub under the vignettes section. So this is the one that we are going to be doing. Now, I've done a bit of cheating. I already exported and compiled this guide, so I'm going to be referring to the HTML document as well, as it's going to make the presentation a bit easier. Whenever I work with reports, which I do often as part of running my company, I like to start with a question. The report needs to be able to answer that question. Think of a report as you would do of an oracle, that you go to a magic oracle in Greek mythology and you need to have a question that you want to ask. Now, this is obviously a tutorial guide that's not meant to be an answering any specific biological question, but it does so nonetheless. If you go to the bottom of the tutorial, the end result here ends up being finding out what types of cells were present in a sample of unlabeled cells. So that is the question that we're going to try to answer with this tutorial. And before we jump into the tutorial itself, I want to try to reverse engineer how we would be able to answer this question. And that's going to make following the tutorial more interesting, in my opinion, and also more clear. What would actually answer that question? Well, you saw it already. It would be a chart showing the types of cells, right? So that's where we want to get to. And we're going to get to that by using cluster analysis, in which we're going to take transcript commonalities and group the cells based on those properties. Now, there are thousands and thousands of different gene expressions in the sample. And to run a cluster analysis, we need a small number of dimensions. And if we have thousands of genes, that means you have thousands of dimensions. That's not really going to work. So we need to reduce the dimensions. And for that, we're going to use a technique called principal component analysis, in which we're going to determine groups of genes that move at the same time in one direction or are reduced in the other direction. And that's going to help us reduce the number of dimensions. Now, to reduce dimensions, we need to make the genes comparable to each other. And in particular, we need to adjust their variance, because some genes have naturally much bigger variance than other genes. And we want to be able to bring them all to the same level. So we're going to scale the data. Now, do we want to scale all data? Probably not, right? If there's a gene that's active in all cells, it's probably not going to help us determine cell type. So we actually only care about the highly variable features. We're going to need to obtain that. And to compare different cells with each other, we're going to need to normalize the data. Are we going to normalize all the data? Probably not. There's going to be junk in there initially, so we need to remove the junk. And of course, if we're going to work with data, we need to load the data. Okay, so these are all of the reverse steps that we're going to take. So we're going to start from the bottom up. We're going to be loading the data, removing the junk, and doing all of those things. Okay, so this is our roadmap. This is our top level view of what's happening. Now let's go and jump into RStudio and go over the tutorial. Okay, we are now in RStudio, and I've gone ahead and created a new folder, which you can call whatever you want. This is going to be our project folder. To create a new project, go to File, New Project, and then select that existing folder that you created, and you will see it here. So this is the folder I created, and I can see now that there's an R project file in there. This is where our project will live. For the tutorial, we're going to need some subfolders, which I'm going to create in Windows here. So it's going to be a folder called Scripts. There's going to be a folder called Data, a folder called Output, and inside of Output, there's going to be a folder called Images. And these folders are referred to in the tutorial, although explicit instructions to create them are not provided, but if you don't have them, it's going to break. The scripts one is one that you can call whatever you want. This is where our main R markdown file is going to live. So back in our studio, I'm going to go inside scripts and click new file, R markdown. And an R markdown file allows us to run R code and also to have documentation, and it's a good medium for learning. When you run this file, you end up with an HTML file each time that you can just double click and open later. All right, so we're going to create this file, and you can call it whatever you want. I'm going to call it tutorial. Cool. So it's created the file. It's got some boilerplate code there. And now we're going to start adding the code from our tutorial. At the start, we have just some boilerplate definitions for the markdown file. There is also some code that will make our save how much time it takes it to run each block. So we're just going to copy all of that as is. It's not really important for the tutorial. All right, so that's the first bit. Next, if we look at it in the guide here, it's going to tell us to download the dataset. So the dataset is a freely available file from 10x Genomics, and it can be found in this link here. So you need to download that, and we're going to unzip it inside of our data folder that we created. Okay, so I've downloaded the file. I'm going to unzip it here, and we're going to see what happens. All right, so it's been unzipped there. I'm going to delete the original file, and next we're going to copy the code to load this file. This is the code for that purpose, and we're going to go over this code to see what it does and what it means. Okay, so the first thing that happens is that it is loading some libraries. We need to make sure that we install the Seurat library in our environment. The other libraries seem to come by default in R, but if not, you also have to install them. I don't remember exactly which ones I had to install, but definitely Seurat was one of them. So for any of these packages, you type here in the console install.packages and then parentheses, double quotes, and the name of the package. In this case, that would be to install Seurat. I have already installed it, and to verify whether you've installed it or whether you've installed it or not, you can go here to packages, and then if you type Seurat, you're going to see which ones you have installed. You don't need to check them in order for it to work in the code, but you do need to check it if you want to be able to run commands here in the console for that library. Also, it helps for the for finding the documentation. So initially, all we're doing is just we're specifying the libraries we're going to use so that they're loaded, and specifying the version of Seurat. Next, we're going to read the file, and here is where we have to be careful with the folder structure to make sure that this matches what's on our computer. For Windows users, notice that this is using the forward slash, not the backward slash that's commonly used in Windows, so just be mindful of that. So let's look in our Explorer to see if this matches. So we have the data folder that we created, but then this is expecting a different file, a different folder name than what I have. The rest seems fine, so that's filter, gene, BC matrices, and then HG19. So that's all good. Just this one needs to be changed. So I'm going to rename it to match what the file is showing. Alternatively, I could modify the file to match my folder structure. But if you don't do that, it's not going to be able to find the file. So the file is being read, and it is being saved in a variable called pbmc.data. For those new to R, and this happened to me, that's why I'm explaining it. In many programming languages, when you use the dot, you're referring to like an attribute of a variable. But in R, this is the same as calling it like underscore. So it's just part of the name. This is a separate name than this one, and it's not like a sub variable or a slot or a attribute or anything like that. All right, so we're loading that file. If you wanted to have a look at it, you can type head parentheses bmc.data, and that's going to show the first few lines. What we do after that is that we create the Seurat object, which is the object that will contain all the data, and this is the object that we are going to be using constantly to work in our project. We're passing in some parameters, so we are saying that that's the data, what we've just loaded. We're giving the project a name. This is completely arbitrary. and specifying other parameters. If you double click on any of these methods and you press F1, this should open the help for that specific entity. It takes a little bit to load, but it's loaded it. So you can see that help on create Seurat object was fine, the following packages. And in here, if you click on any of these, it will give you documentation and more information on the parameters that it's using. Okay, so it's important to do this from the beginning and make sure that you know what each thing means. All right, so next we're just displaying that on the screen to see what it is. So I'm gonna close that code block there, and we're gonna knit. Knit is essentially executing this. It's going to compile it into an HTML document. So let's click that. Okay, after rendering, and by the way, rendering takes a few seconds and it can even take minutes depending on what you're rendering. We can see now the output. So you can see the code that we executed. You can see that this is what the data looks like. Each row is labeled by a gene, and each column represents a different cell. These are essentially 2,700 cells represented in this matrix. And if we scroll down, when we output the PBMC, the Seurat object, we can see some information about it so that we know that 13,714 different genes are expressed across 2,700 samples with one assay. So this gives you a bit of a summary of the file. Next in the tutorial, we start with the pre-processing workflow. Remember that I said that we're going to get rid of the junk. And the junk here, there's not a clear definition for it. It depends on what you're doing, what you're after. In this tutorial, they mentioned that they want to get rid of cells that have too much mitochondrial expression. And for that, they're adding a calculation that adds a percentage of the genes that correspond to mitochondrial gene, and they all have the MT pattern in their name. So this code will just add extra information for each cell in terms of what percentage of mitochondria is present. So we're going to copy that into our code, and we can visualize that using violin plots, which you can see here. What other things are going to be discarded? We're going to discard cells that have too many genes expressions, and we're also going to get cells that have too little. What does each thing mean from a biological sense? I don't have that intuition because, as I mentioned, I'm only just starting out this journey. From a quick read, it seemed like if there are just too many genes being expressed there, it could represent that more than one cell is being the two cells were added together in one entry. So that's not really a valid entry. And I'm not too sure what it would mean that a cell has just too little expression. But essentially, these three charts were generated, and based on these three charts plus the criteria of somebody with experience in this field would make it possible to define some cutoff points. They also plotted together some of these elements with each other. So how many different RNA genes were found, how many in total, and the mitochondrial percentage. So we're going to copy that into our file. And from there, they kind of decided that they're getting rid of they're only keeping the entries that have at least 200 different genes and less than 2,500 genes and a mitochondrial percentage of less than 5%. Please let me know in the comments, what are your thoughts on this? Is this a common setup, or is this like an arbitrary thing, or does it change in each experiment? Regardless, we're going to run this just to see if we got everything running on our end. Okay, so everything seems to be running okay. I've got all the different charts here, and we're ready to continue on. After getting rid of all the junk, we want to normalize the data so that cells are comparable with each other. And you can see here that it is showing lots of default parameters here for the normalized data function. But if you omit them, that's the same thing. So in this tutorial, it will often show you parameters only to mention that those are the default ones. So I'm going to copy that into our code. And now that we've normalized the data, we want to try to find which genes have a highly variable expression across cells. Because think about it, if a gene is expressed exactly the same across all the cells, it's not really going to help us differentiate cell types because it's going to be the same in all cells, right? So we want to find the ones that do have that high variation, and that is what this code is doing. It is running a find variable features, specifying like the algorithm used and how many are we going to find. So which ones are the top 2,000 out of 13,000 that we had initially. We're going to end up with only 2,000 for the subsequent analysis. We're also identifying the top 10 ones, and we're running a bunch of different plots. We are being explicit as to which part of the Seurat object is going to be used for this calculation. Although it's my understanding that this is not necessary, that it would happen by default. And to be completely honest, I don't really understand exactly what we're doing here with this counts string. But let's go and select it and paste it here, and let's run it to make sure we're all good. Okay, so you can see here it is showing us the variable feature plot, and it's saying like, see, the ones that have the least variance are the black ones, and those are kicked out for future analysis, and we're only focusing on the top 2,000 ones. And then it's displaying the label ones because this might already tell you important information about the sample. Next, we're going to scale the data, and that means we're going to bring the variance of each gene to 1 across all cells, so that each gene would weigh the same in the subsequent analysis. Otherwise, you could have one gene that could like dominate too much, and that's not what we want for the purpose of this analysis. So I'm going to copy this code that will essentially run that scaling the data, and it saves it back into the Seurat object. Next, we're going to do a linear dimension reduction, and I mentioned this earlier. Okay, so we had 13,700 genes. We reduced that to 2,000 genes, right, with our variable feature step. But 2,000 dimensions, I mean, we can barely think in 3 dimensions. I mean, some people can't really think in 3 dimensions. It is hard for humans to think in more than a handful of dimensions, right? I mean, try to think of a 5-dimensional space and things get tricky. So for any kind of clustering, we need less dimensions. And what we're doing here is creating principal components. And how can we think about them, all right? Think of them as a vector that in one direction, all of these genes, when one of these genes increases, you see all of these other genes increasing as well, and you see all of these other genes decreasing. When these genes here increase, or any of these things increase, you see the other ones in the group also increasing, and the ones in the other set decreasing. So that would be one principal component. So you find multiple of these quote unquote like directions in this multi-dimensional space, and you make those the dimensions for further analysis. Because if all of these genes move at the same time, you can kind of treat it as one in this type of framework. So we're gonna copy this code to run the PCA, and we can examine the first 5, and note that more than 5 are created. I think it is 50, and I don't know what the default is for the number of features. We're only showing the top 5, the most important ones, but there would certainly be more, and I'm not familiar with the exact number. Perhaps if you know, you can add it in the comments, and we can all learn from that. And we can visualize this in different ways. For example, with vis dim loading, and we're just showing the first 2 PCs. You can see the genes that have the most presence here, and you can see the value of that dimension. I do find it interesting that a lot of them it's like a more I wonder, for people with experience in the field, if the fact that's almost like a vertical line might mean something as opposed to like a more horizontal line. I imagine there could be some significance to that, but I don't have the intuition to answer that question. Also, they can be plotted against each other, and I presume that these could be able to detect kind of like get an idea of the clusters or what that might indicate. This potentially might indicate that there's some independence from each other. For example, there is at a kind of narrow range of PC1, there's a big variety in PC2, and for this cluster here, it's sort of the opposite. But I don't have the intuition to say something useful about this plot. And then we have the heat maps. So these are very important plots in bioinformatics. Yellow means high expression of a gene, purple means low expression of a gene. each column represents a cell in the sample. And you can see that, for example, for that little cell there, it looks like there's a high expression of MALAT1, but then there could be, a lower expression of some other gene. And how clean this looks indicates how strong this PC might be. Because if it's all random, if it looks random, it's probably not very, it's more noise and signal. In here, they plot all of the different ones, or the first 15, and you can kind of see that pattern where initially they're more clear, so you might be able to derive something biological from it, but the further you go, the more kind of messy it becomes. And this is determining the dimensionality. Okay, so we have created these PCs, but how many of them matter, right? We went from 13,000 to 2,000, and now we've got 15 or up to 50 were generated, but we still don't want so many. And it says here, okay, so how many do we actually need? That's more of a judgment call. And it says here that they use an oboe plot to determine that. This plot ranks the components based on the percentage of variance explained by each one. So you can see that some of them explain a lot of the variance, but some of them not as much. Where exactly do you draw the line? It's not only from the plot. It says here that there are also biological considerations that might be relevant for the particular experiment that you're doing. For example, if you care about these specific cells, they seem to be strong on PCs 12 and 13. So maybe you should include those if you care about those cells. This certainly seems like something that requires intuition and experience because it's not just a clear cut like other things that you might encounter. So we're going to copy that into our code and continue our tutorial. Up to this point, we've done a lot, so I'm going to make sure that we're still executing everything. And I like to do this in general so that I can follow along. And I'm just to make sure that I can run it on my computer. There's always a lot of safety when you get to that point. All right, so we've executed here the code, and we have our principal component related charts. We've got all the cool plots as well as the elbow plot. So we are good to continue. If we go back to our roadmap, we've done all of these steps. We've gone through so many steps to get to this point, and now we're pretty much on the final stretch with the cluster analysis, and then we're ready to answer the question that we set out to respond. So clustering, there are a lot of different clustering algorithms. It looks like there are good defaults here that probably do the job. Specifically, there is a call to find neighbors first, and it's taking only the first 10 dimensions because I forgot to mention that we chose 10 of those principal components. So we're running the neighbors just for those 10, and then we're finding the clusters. So let's add that to our code. So this is going to give us an output here. It's running the algorithm. This can take a while, actually. And once we have the clusters, okay, the clusters are in a 10-dimensional space. So imagine a 10-dimensional space. I'm sure that you can do it. Our clusters are grouped into that space. But now we want to project that 10-dimensional space into a 2D surface that we as humans can easily process. And that's what UMAP does. It takes that 10-dimensional space and projects it into a boring 2D figure. All right, so I'm sure we've lost a lot of richness from this process. I mean, nothing comes for free, but we get something like this, and that looks like something interesting that we might care about. So there are options here to save the data as well. And we're going to add that and then check if it actually got saved. But we want to see now, okay, what's actually the difference between each cluster? What biomarkers are expressed are the most meaningful within that cluster? And you can run a few different like queries here. I like to think them as queries because it's almost like you're inquiring for different things. For example, here you're finding all the markers for cluster 2, and you write it, you pass in the Seurat object, and you pass the identifier of the cluster, and then there's a minimum cutout point for which those markers matter. So then we get those 5 genes for that cluster. But you can also might want to find the ones that are distinguishing cluster 5 from cluster 0 and 3. So you can kind of run this like query, like SQL kind of thinking type of language. And that's going to depend on what you actually want to answer. You can also find markers for every cluster compared to all remaining cells and report only positive. So that's like a more complex, a bit of like a nested type of query. And you can also like choose to visualize like specific genes, like how they, for example, this particular gene, like where is it located the most in which ones of the clusters. In this case, it would be a cluster 5, and it kind of tells you a distribution of the expression level. So I don't want to copy all of this because it's gonna take a little bit too long to run, but I did want to show you just this kind of stuff that you can do. And these are things that you often see on papers. And at least for me, that I'm a total newbie into all of these things, it's been an eye-opener after running all of these tutorials and then just looking at papers, and I'm like, oh, I can actually know how that was generated. Here we're looking, for instance, at a bunch of genes, and we want to see like how they're expressed across different clusters. So that could be interesting for some analysis, and a heat map as well. But this is how we actually respond to our biological question. We want to know the types of cells. And for that, we need to consult, I guess, external libraries or people that know this from experience, relate what markers are characteristic of which types of cell. I obviously don't know that information by heart, but this is, once you have this information, which I assume is obtained from biological databases, and please let me know in the comments if you know how this is obtained, then you can easily like create those IDs per cluster. So you're giving them each an ID, and you can finally visualize the results. So this is one that I do want to have in my own file, and we're gonna go and execute that. All right. Okay, so we have now the final chart. We know what types of cells are present in this sample. We were also able to save that file. So let's check our file explorer. Inside of output images, we should have that image saved as well. So with that, we're gonna conclude our tutorial. If you have any comments, if you have any questions, if I made big mistakes, please let me know in the comments. I'm learning in public. I'm not an expert in this. I'm absolutely obsessed with biology, and now I'm kind of focusing on connecting that with the software world that I so much love and care for. So this is just the first step here, and I look forward to seeing you in the next video.